So I'm going to talk a little bit about the early life exposure to air pollution and mainly the black carbon and health outcomes, which is mainly mediated by the extracellular vesicles. So I will mainly focus on that part. For introduction part, I should say that I've done my bachelor in the cellular and molecular biology. Then I had a master of the human genetics, and I have a medical PhD in the molecular medicine, but all from Iran. And then now I'm a PhD candidate, both in Hasselt University and in Maastricht University. You all know the Iran with the Persian cat, caviar, oil wells, rocks. I don't have any of them here, so I cannot offer anything. But what is toxicogenomics? It's clearly the effect of the toxins on the genome. So it's very clear. After the discussion we had today and the day before, Monday, so now we know that what is it. But exactly what we are looking in the toxicogenomics is the changes that are made on the DNA. Mainly we can track them as a genomics changes or epigenomic changes. Then what is happening on the transcriptomics world, which is very important in the complicated diseases, and then in a combination with proteomics and metabolomics, we can get lots of information from the exposure data that we can collect for the exposures, of course. And mainly we have the bioinformatics and system biology for the predictive toxicology beside all these things. So what toxicogenomics offers to us, what exactly and how we can use it. So mainly for understanding the environmental stress in inducing the diseases, mainly the complicated diseases such as the cancers, autoimmune diseases, which are mainly multifactorial in genetic basis or beyond the genetic basis. So we can then understand the molecular mechanisms based on the toxicity, and then we can introduce some kind of biomarkers, which are mainly very different and varied, that as Stefano explained, that we can use them for different aims, such as the diagnostic, prognostic, predictive, or risk assessment, or monitoring, or lots of other things that we can include here. So I'm going to explain a little bit about my own project that I'm doing, so it will be more in practice, so that I hope you find it interesting. What we know from the early life exposure, that early life exposure can change the function of the cells mainly, and main organs that they can influence our development. So the main mechanisms which are introduced are inflammation and also the oxidative stress. So we know that they can change mainly lots of things in the DNA, in the RNA, in the protein level, in the metabolites level. So we all are aware of this part. So we can track the changes from the prenatal to the postnatal exposures. But the point is that we know normally when we are talking about the prenatal exposure, we only include the mother and the fetus. But I want to include the father for the first time, because we know that also the air pollution mainly can affect the sperm quality. Maybe you will ask how. I will just look at everything from the extracellular vesicle aspects. There is a report recently that we know that the environmental stresses, they can induce certain extracellular vesicles, that they can change the metabolism status of the sperm through delivering the specific miRNAs. So we all know that miRNAs are very good tools to change or shift the pathways inside the cells. So we could track this. So now we know that the changes that are happening as acute exposure or as a chronic exposure, they can change the sperm quality. So those changes can be influence the development of the fetus also. So as also Nelly mentioned, that 99% of the global population is exposed to the air pollution higher than the level of the WHO. I don't know where those 1% live. I'm so curious about that part. And we know that normally they say that 1/5 of the global burden of the newborn health disorders are associated with the air pollution. So some of it is understandable. But in the term of the air pollution, we know that we have different sources that we can divide them to indoor, outdoor, natural, or even we can make them as a category of industrial. But what I'm going to talk mainly is not the gases, not the liquids, mainly about the particles and particulate matters. So if we take a close look to them, we know that they are divided to 3 categories. So by now we all are familiar with this part. But what is the difference? The difference between them is like the penetration capability, that they can induce different effects. The first one, the PM10s, they are not that much strong to penetrate to the lower respiratory system, so they are mainly trapped on the upper respiratory system. And the second group are the PM2.5. They also can go farther, but the effect that they can make is normally higher than the fine particles. However, from the fine particles, we expect a systematic response. But what they suggest, because they are very small, the Clarion system can normally get them out of the body, or the immune system can help for the precipitation and removing them. But the effects of the 2.5 particulate matters are more long-lasting. In the term of the particular matters, mainly from the black carbon aspect, we know that they are mainly because of the incomplete combustion of the fuel they are produced. But the good point is that they are always correlated with the carbon dioxide, monoxide, and VOCs. So somehow we can track them. So if we can track one of them, we will be able to analyze that the exposure level is correct or not. In the size, for the black carbon size, there are different literatures that are reporting different sizes. It's not exactly clear by now that how big are they, but what is assumed is something between the 50 nanometer to thousands of the nanometer for these particles. And mainly the absorption or the entrance of them to the body is mainly by the inhalation or by the eating the polluted drinks or foods that are polluted by the black carbon. And also there are some reports that in high doses they can enter to the body from the skin, but of course mainly it should be followed by the occupational exposures. So the systemic effect of the air pollution can be tracked on the brain and the heart and almost all the organs in the adults. But the point is that in the adults, the immune system can make a clearance of these particles. So somehow we adapt our body to these exposures. But what will happen during the pregnancy? Because we know that the immune system of the fetus is not that much strong. Still, it's dependent on the mother immune system. So what will happen if they reach there? What they can cause? So mainly what we know, they can cause the growth and the development, the growth, allergic diseases, neurodevelopmental diseases, but this is something that we can see after the birth. So these are postnatal effects. So if you take a closer look, you will see that in the different weeks of the pregnancy, the effect of the air pollution is different. For example, from the 3rd week to the normally the 3rd semester, you can see that interrupted placental development and fetal growth restriction, reduced weight, and also we have the early susceptibility to the later preterm birth or the preterm birth or immature babies, and also we have the heart defects. And I want to drive your attention to the CNS development in the fetus, because I will talk a little bit about this part, and you see that it's normally happening on the late pregnancy. So this was a paper I found it very interesting, because they are elucidating the effect of the air pollution for the different particles from PM 2.5, NO2, ozone, and PM10 by showing the pathways that they are interfering. For example, the orange one is the DNA methylation, the blue one is RNA expression, and microRNAs are the purple one, and the green one are the non-coding RNAs. If I'm not colorblind, I should say the colors correctly. Yeah. So we can see the different effects: preterm birth, birth weight, fetal growth, growth restriction, newborn neurobehavior, and lots of spectrum of the diseases which are mainly connected to the brain development. If we take a closer look to the non-coding RNAs world, we will see that we have lots of categories about the non-coding RNAs. Why they are important? I will explain it in the next slides, that why I'm focusing on the non-coding RNAs, because normally what we can track on the DNA, somehow it's clear what effect we will see. But in the RNA world, it's not really clear that what we are looking for and what we expect. But maybe you have heard nowadays that we are mainly focusing on the macroRNAs or microRNAs or MERS. And I want just to add this point that the combination of the microRNAs and also the circular RNAs from the long non-coding RNAs is very important, because we always look at the microRNAs. We know that they're just somehow regulating the expression levels of the gene negatively, so they're correlated together negatively. But what controls the mRNA expression? So the answer is here, long non-coding RNAs, mainly the circular ones and non-circular ones. They're working as a sponge inside the cell. They can absorb lots of the microRNAs at the time, so they can release them and they can shift the pathways inside the cell. So we can see lots of the changes in the pathway shifts just only because of presence of the long non-coding RNAs. So this part is very important in my project. If we take a close look to the placenta function, I think you all are familiar with it. So protecting the baby, production of the hormones, modulating the immune responses, oxygen, nutrition, removing the wastes, and all of these things. But what will happen if they are exposed to the air pollution? I'm mainly showing the PM 2.5 because most of the studies are conducted on this part. So what we are expecting to see, electrophilic intermediates that will change the function of the placenta, redox signaling, inflammatory response, altered metabolic status. Also, we have the chromatin remodeling and altered nucleosome positioning and transcriptional regulation that all are correlated to the certain diseases. This part Nelly also explained that we have, in our department, we have seen that black carbon can reach to the fetus part of the placenta. So from there, it can reach to the cord blood, and from there, it can reach to the fetus directly. And thanks to the studies from the EVA, EVA showed that there is a correlation between the presence of the black carbons inside the fetus organs. Of course, these are mainly from the aborted childs, and you can see, but still there is a big correlation between the presence of the black carbons in the placenta and presence of the black carbon in the fetus. So it shows that the higher we are exposed, the higher accumulation of in the placenta, then we have a higher amount of the black carbon inside the fetus. So it's very clear, it's a direct correlation. Let's take a closer look to the placenta. What's happening inside the placenta? So the main part of the placenta, or mainly the last layer of the placenta, which is connecting the mother and the fetus, are the trophoblast cells. So we know that placenta is a fetus organ mainly. So we will focus on the trophoblast because, as we see, that most of the things are adjusted, controlling, and modulating by the trophoblast cells. Normally, we have three categories of the extracellular, trophoblasts. We have the extravenous trophoblasts, which are directly contacting with the nutrition or whatever are there, the CO2, oxygen, whatever that you can see there. And also we have the syncytotrophoblasts and the cytotrophoblasts. But why it is important? For my project, this is very important because they are responsible for producing or releasing most of the extracellular vesicles inside the cord blood. So it means that they can produce this exosome. So the exosomes are presenting there, so we can follow them. As Stefano also explained, mainly the biomarkers are not that much handy because of the invasiveness. So we are developing non-invasive biomarkers. So we are trying to do something that make it non-invasive, to see that how we can follow it. So the presence of the extracellular vesicles from the placenta can be tracked inside the cord blood or inside the mother blood. But what are the extracellular vesicles that I'm talking about them? In general, we have three categories of three classes of the extracellular vesicles based on their size and their release. They're different. The smallest ones are the exosomes. This is the name before the 2023. Now they call it small extracellular vesicles, but let me say exosomes, so it will be easier. Then we have the microvesicles from 100 to 1000 nanometer, and finally we have the apoptotic bodies. If you notice, you will see that the mechanism of the release is also different, the exocytous, budding, and blebbing. For the budding and blebbing, it's normally by chance. There is no loading mechanism of them. But for the exosomes, there is a loading mechanism. And they are smaller than the 200 nanometer, which is mainly the BBB, blood-brain barriers that they can screen. So if there is an exosome from your brain or from your liver, we can detect it in your saliva, and your urine, and your blood, and all the body fluids that we can collect from a patient or from someone, volunteer, so we can track them. So this is very important. If we take a closer look to the biogenesis of these particles or these microvesicles, you will see that the loading is not random. The loading is based on the specific mechanism that we call it ESCRT. That is the full explanation of the SCRT. It means that cells, they use them to load with specific molecules or specific biomolecules to communicate with each other. The effect can be inside or the effect can be removed. So these exosomes can reach to the target cells specifically. For example, if we are using them for the treatment, for example, for a drug delivery, if you take exosomes from the breast fluids, so they will specifically go to the breast cells because there are some integrin patterns on them that they can recognize each other. I mean the original tissue. So those are very important for the delivering the messages. So nowadays they are known as the third-party communicating. But why extracellular vesicles? Why they are important? Because normally we have seen that they are overexpressed in the pathological conditions. Why overexpression is important? If we notice the limitations of the techniques that we are using now, you will prefer to detect something that is overexpressed or downregulated. So normally overexpression is easier to detect. Imagine we have a gene, the expression is lower. So in your qPCR or your real-time PCR, you need more cycles, so it is error prone. But if it is overexpressed, so detection is easier, faster. So it will be better for you to work with the biomarkers that are overexpressed. And normally if you see in your normal blood tests that you get from your doctor, almost everything is overexpressed if you are looking for pathological conditions. We rarely have some biomarkers in the clinic that we are looking for the downregulation of them. So next one is the nanoscale of these vesicles. So they can go through all the barriers, as I explained. If we are going to study, for example, the liver, we don't need to get the biopsy directly. We can collect the exosomes from the blood. Also, they can mirror the functional status of the original cell. So accessing to the brain cells is so hard, but we can isolate them. We can isolate the extracellular vesicles from the brain, so they will mirror what is happening in the brain. Also, they can preserve their cargo from degradation. The half-life of the RNA in the normal sampling is 7 to 8 hours. But in the exosomes, if you leave the exosome with the RNA inside it on the table, it will be stable for 5 years. So that's very good. Also, the neutralization from the blood is not that much, that we can keep them for a longer time. So what is inside the exosomes? 76% are microRNAs. So it shows that they are using for the communication with the cells for certain changes in real time. So those are very important for communicating within the cells or inside the cells. That's why I said that we are focusing mainly on the microRNAs. But there is also another advantage, is that what we are focusing on the exosomal microRNAs in contrast to the cell-free microRNAs. Cell-free microRNAs is what when you're collecting the blood and you're directly going for isolating the RNA from the blood and then you study it. We call them cell-free mRNAs. Cell-free mRNAs are normally not that much stable because our blood is full of the exogenous RNases. And also they can easily be disrupted, dysregulated by food that you eat, by drink that you drink, by sport, by hypoxia, by infection. So lots of things they can influence their level. But what is inside the exosomes are not that much prone to error. And also, sometimes you find the biomarkers from the blood, from the cell-free biomarkers, but they are not correlated exactly to the original pathologic tissue. What is happening is that sometimes that we detect the response of the body to that pathological condition. So this is also an important point. And in most of the cell-free mRNA researchers from the different labs, there is no concordance. It means that different research groups are reporting this, but normally you cannot repeat it because of the techniques, because of the sampling, because of the preserving that they are using. But what we are doing here, as also Nelly explained, we have a very good cohort here that we are following it. And last month we had a pizza party for the 2000 sample collection from here. But you can see here that what kind of information we are collecting from the participants. So normally we have almost everything that we want to study on them. We have the placenta, cord blood, meconium, also maternal blood, urine, vocal cell, saliva, nail, blood, urine, maternal blood. So it is very comprehensive biobank that almost we can access to lots of samples. But in general, in a snapshot, I'm working on two cohorts, one from the environment from the Belgium and another one from the child with cancer. It's from the UK, from the Bradford and Manchester University. So what we are exactly doing, we are measuring the black carbon exposure inside the mother, then we do it in the placenta, in the placental tissue, mainly the trophoblast, because we are looking for those cells, and then we are trying to indirectly develop a method that we can see that what's going inside the fetus brain. Imagine you want to assess the function of the cells inside the fetus, which is inside the womb of the mother. So normally we only have the imaging, but it's not that much strong. So what we are going to do, we are going first to take the tissue from the placenta, from the biobank. Then we will get the exosomes. As you can see here, I don't know if these are the specifically placental exosomes. I will explain how we are going to isolate them. Then we have the fetus brain exosomes. We're going to isolate the fetus brain exosomes. This is the non-invasive biomarker that we are going to isolate the exosomes from the brain of the fetus inside the womb of the mother. So this is hard enough, I think, but I think it costs to track them. Also, we will measure the effect of the exposure to the black carbon in the whole blood cells inside the cord blood, then on the specific exosomes from the placenta and fetus brain, and also on the general exosomes to see that how they can change their functions. We have some data from the modeled measurements. So somehow we just use this for the screening. Then after the screening, as Nelly also explained, we will do the direct measurement. So then we are assured that how much we have there, because we want to correlate it to the molecular changes. So, but how we isolate the trophoblast? As I showed you, the trophoblast is the last layer of the placenta. So we use the biopsies from the biobank. So we make slides from them, and then we do a specific toluene and blue staining, because this trophoblast, they contain lots of nucleuses, so they are acidic, so we can stain them. After the staining, we will see something like this under the light microscopy. But then what we do, so this is a LCM, or laser capture microdissection. After we stain the cells on the tissue, so we specifically choose the parts that we want. So we exactly isolate the cells that we need. So we take them and we use them for the RNA isolation. So now somehow we are assured that with the high percentage of the cells that we are working on them are trophoblasts specifically. So then these are the pieces that you can see under the microscopy after the isolation with the microdissection. So normally we will perform the RNA-seq to analyze the data. But how we isolate the exosomes? For the general isolation, we use the combination of the three methods to sacrifice the yield for the specificity. So we combine the precipitation, serial centrifugation, and also size exclusion chromatography. Then we have a very high pure exosomes, because we are looking for biomarkers. So we want to be assured that they're really there. But how we isolate the placental exosomes and fetus brain exosomes? So this is the challenging part, that we found a specific molecule known as a plug that are existing only on the placental cell. So we are using this marker to track the exosomes from the placenta. And for the brain fetus, we figured out that there is a marker called TAG1. It is specifically and transiently expressed during the fetus brain development. So we specifically isolate the exosomes from the fetus, not from the adults. So the cells from the mother, they wouldn't interfere with our method. How we do this? Very easy. We have a microbeads. We use the specific antibodies against those antigens that we have. Then we cook them with the microvesicles. And after the isolation with the size exclusion chromatography, then we will use them, or we are using them to specifically isolate our tissue-specific exosomes. For the confirming, we have the size of the microvesicles. We should confirm them with their size, and then we should confirm them with the electron microscopy, mainly the cryo-TEM. Then we need to confirm their shape, and then we need to confirm them with the Western blotting for the specific markers, because these exosomes, they carry specific markers. We cannot call every particle exosomes. They should show this characteristic. So we need all the ticks in our study. This is the nanoparticle tracking part. It's also a very short movie that we are using the exosomes to pass through the camera. Then we are capturing them to specifically make their size. Then you can see that how they are accumulated there. Then it will show us the size distribution. So somehow we are assured that what we are doing is in our range. Then this is something new that I'm going. This is very new results we can get. We can detect the microvesicles as a carriers for the black carbon. So we figured out that black carbon is which is accumulated inside the placenta. We isolated the specific exosomes from them, and then we noticed that this exosome carry the black carbon. The higher amount of the black carbon inside the placenta, the higher amount of the black carbon inside the exosomes. So how we did this, we isolated the exosomes, then we stained them with the specific stain, and then we follow them by the technique that also Nelly explained. So we use the Z-stack, so we confirmed it. Then we use the colocalization to be assured that everything is inside. Not the exosome is here, the black carbon is here, so it's a false data. No, everything is inside. So you can see somehow here, you cannot see, okay? There are some red dots on the left, there are some red green dots on the right. They are matching together. So this is the results. What we figured out from our pilot study, we figured out that 65% of the black carbon is carried by the microvesicles. So that is a very significant mechanism for transferring of the black carbon from the placenta to the fetus. So what we finally do, we will use this specific exosomes to perform the RNA-seq. Then we can realize that what's happening inside the tissues that we are targeting, and then we can correlate the specific tissues with the specific microRNAs inside the exosomes. So somehow we are developing non-invasive biomarkers based on the microRNAs. And the last point is that the signature of the microRNAs, they are the best combination to detect the origin of the tissues. We can do it, for example, with the mRNA, but that is not that much specific. The best signatures or profiles that they can use is the combination of the microRNAs together, so they will give you an extra validation that's what you are going to do. So these are the conclusion that how you can use the toxicogenomics thanks to ChatGPT 4+. And I didn't pay for it. We have a subscription from university. So anyway, and thank you. This is a lake from my hometown, but I want to mention something that this is a salty lake. And nowadays, because of the global changing, we are noticing that we are exposing ourselves to new pollutants. What is inside the oceans or inside the seas or inside the lake, that the weather is changing. So we are exposing ourselves to new pollutants. So maybe it's a kind of warning for us to be assured that what's happening around us. Thank you.